online. Reed is online. Tutor is online. All right, let's see. Do we have... Wait, Burdette is online. Anyway, do we have it? Hudson's here. Newton. Massey. Richardson. Cedric. Rice is online. Paulie. Eric. Morning. Richardson, right? David. Kip. Let's see. Asbury is here in red. A little extra credit. Bowers. Hemmings. Redmond. Reed. Michaela's here in red. Taylor. Yep, you're here, right? Yep, you are here. But not in red. That's what I was trying to confirm with you. Van Cross. All right, well, we still have a little time. Actually, I wasn't recording. No, I am recording. Okay. Let me give it a second. Does anybody have any questions before I do announcements? Because the announcements can be pretty important, so I want to make sure this one's done at 8 instead of early. I want to make sure everyone hears it. Then again, I'm kind of preaching to the choir because it's only important for the people who missed lab this week. And most likely, if you missed lab this week, you're here and present and paying attention. Or if you didn't miss lab this week, you are here and present and paying attention. Anyway, it's 8 o'clock. So I guess for attendance, I'll remind you normal stuff. If you're in person, send me the words before 9 a.m. for extra credit if you want. If you're online, send me the words before 9 a.m. for regular attendance. And if you're watching the video, we'll see. I might have questions to go along with the video, or you might just follow the instructions that I give during the video. So that being said, the first word will be different for everybody. So instead of a word, send your birthday. And if you're paranoid about people having that kind of stuff for identity theft reasons, you can just give me the birthday and leave out the year or whatever. I might have access to it anyway. I don't know. But yes, so the first attendance word, if you will, is actually your birthday. So send me your birthday. All right, it is 8:01, so let's do announcements. And this is mostly, again, I'm probably preaching to the choir because most likely the people who need to hear this are not hearing this right now. And hopefully they will hear it when they see the video. I see somebody left the meeting. And we have someone here. Massey is here. Anyway, if you missed lab this week, for now, I'm still trying to figure out what to do because you can't miss lab. I've said that since the beginning. I'm really loosey-goosey with lecture, right? Because you can watch a video or stream online. But with lab, you have to be there. And you need to be on time, right? So if you miss lab this week, I'm still trying to figure out exactly what to do. But for now, watch the video I've already posted, and you'll get 1 quarter credit, right? And I'm going to figure out what else you can do to get the other quarter, right? So you can at least get half credit. But here's some advice, and this isn't just for my class, but this is for your entire college career and probably your whole life. If you don't know what to do, take some damn initiative. Pardon the French. But obviously, I've been posting that video for you guys to do it, and you didn't like, oh, I couldn't come to lab or whatever your reason. You should have some initiative and say, obviously, I need to watch this video and take these questions. Maybe I'm not going to get any credit for it, but this video explains what we're going to do in lab next week, right? So at the very least, you should have done it just because you want to learn it and you need to learn it so you know what you're doing in lab and you know how to write a lab report, right? So at the very least, you could have done that. But also, it will help you in life when you want to get stuff done. When you want to ask somebody for something, just say, look, I know you didn't ask me to do this, but I went ahead and did this. So is there any way I can get this, right? Take Take control, take charge, get stuff done. Don't wait for me to tell you you have to do it, especially when it's obvious that it needs to be done. Anyway, so that's what's going on. If you miss lab this Wednesday or this week, watch those videos, and I'm going to post this all in writing. Watch those videos. You'll get at least quarter credit for what lab last week. Extra stuff you can do to get the other quarter credit. So, are there any questions? Of course, if you're online, I can't see. All right, let's move forward then. Sorry to start off on a sour note, but labs are important, and especially this one because you got 20 points just for showing up for the pre-lab this week, 20 points for doing the lab next week, and then 20 points for the lab report. So it's worth 60 points. It's pretty hefty. Here we go. And while this is loading, it's also a good time to remind you, if you don't like the grades you've gotten on labs or you've completely missed labs before, it's not too late. It's never too late. Just go back and fix mistakes you made or turn in labs late. It's better to get half credit or partial credit than no credit. And as always, I'm here to help you. You don't have to do it alone. Even if you just didn't do a lab and you're like, I didn't come, I was sleeping in, which is horrible. But even then, I will still help you. I will meet you online and walk you through and tell you what the experiments were like and how we got the data and all that fun stuff. I'm here to help you. Anyway, let's jump back into respiration. This is actually electron transport chain. This is where we left off. Let's back off. Let me back up a little bit and talk about this. The last thing we talked about was this whole flow of electrons, right? So to remind you, a lot of the exam is going to be in which stage does this happen? In which stage does that happen? In which stage is this used? In which stage is that produced, right? Most of the questions for respiration are going to be like that. But there are going to be a few questions that deal with like just the flow of electrons. Where do they come from? Where do they go? And I saw finally yesterday, I was looking at my emails and looking at the chat. I can't see when you guys chat online, but somebody said last time after I said, where did you come from? Where did you go? Somebody typed Cotton Eye Joe, and that made my day yesterday. Anyway, so yes, you need to know where the electrons came from, which is glucose. And where did they go? They go to oxygen. What happens when the oxygen gets that electron? It forms water, right? So that's where we left off. Know what happens with the electrons. And again, for this picture, I talked about the fact that you need to know that this is the molecule, the NADH. That's the thing that's moving the electrons around. I told you that we're going to talk about this later, which will be today, right? How do we make ATP from these electrons giving off energy? I also talked about the fact that, yes, the electrons go to the oxygen and that forms water, but technically you need something else, right? So technically you need two hydrogen ions and 2 electrons to make that happen. But you don't need to worry about those details. So again, the fact is that we are stripping electrons away from glucose, and then they go down a trip, right, releasing their energy. Oxygen finally accepts them, and then it forms water. So any questions about that side? Okay, transport chain. Right, so let me back up again. Sorry. These things right here, these proteins, because this is just a schematic, right? This picture right here, it doesn't actually show you where things are happening. It's just kind of nice to show that, you start with high energy, right? Because those are high energy electrons, and then they go down, they literally go down to a stable state. So high energy to low energy, it's giving off energy. That's why it was illustrated that way. But it doesn't show you the electron transfer, the mitochondria. So you need to know, first of all, that this is happening in the mitochondria. That's definitely going to be a test question. Which of the 3 stages happens in the mitochondria? And the 2 answers are the electron transport chain and the citric acid. But in this case, it's a little bit more important. As far as we're concerned, the fact that the citric acid cycle happens inside the mitochondria, that's just trivial information that I'm asking you to know. In this case, we can't have as much ATP as we have without the fact that it's in the mitochondria. We're going back to that form and function thing here. The shape and the form of the mitochondria lends itself to what it's doing. Because the membranes are highly folded, first of all. So how do I explain this? I wish I brought some trash bags. So imagine the mitochondria is almost like a big trash bag, but inside of it is an even bigger trash bag, right? Or no, I shouldn't say trash bag. So maybe the outside of the mitochondria, the outer membrane, which is like our cell membrane, it's like a Kroger bag. Can you imagine that? Just a small Kroger bag. And then on the inside, you take a whole big garbage bag and you've crumbled it up and you've now fit it into that smaller bag. Can you guys picture that? Hopefully you can. So the outer membrane is nice and smooth-ish, right? Because it's just the Kroger bag and it's stuffed with that bigger bag. And the bigger bag, like to get it in there, I had to squish it together, right? So it's big. It has a lot of surface area. It just happens to be squished in that little Kroger bag. Same thing here. So we've got that outer membrane for the mitochondria. And then we have yet another membrane on the inside. So unlike our cells, right? Our cells just have that the one membrane. But these things have two membranes. There's the one on the outside, one on the inside. The one on the inside is really squished together and folded. And that's important because it has a large surface area. Therefore, it has thousands of copies of electron transport chain. So again, imagine you had the Kroger bag versus the garbage bag. And imagine I said, all right, I want you to put a dot every 3 inches apart on these bags, right? And you have that little Kroger bag, and you have the big garbage bag, and you've drawn dots every 3 inches apart. Which one's going to have more dots? The larger bag, right? Because it has a larger surface area. Same thing here. So we've got that larger bag squished up, has a lot of surface area. Therefore, it has a lot of those proteins. These proteins right here, those purple things. And those proteins are the things that are actually making the ATP. And this is what it's all about, right? This whole process, all of cellular respiration. The first two stages is all about stripping away high energy electrons so that they can be used in the electron transport chain. I keep saying that. And now I'm coming back to it and saying all those high energy electrons that we stripped away, they're being put to use by these proteins. And those are the things that are making the ATP, the most of the ATP. Because remember, all three stages produce a little bit of ATP. But it's this third one that produces the most. It's all about those high energy electrons. So the next slide I'm going to show you is an illustration of this. So again, the previous slide was more of a schematic that shows, hey, we start at high energy and we go to low energy, and the electrons give off energy and we make ATP, all that stuff. But the next one's actually going to show more of what it looks like at the cellular level or molecular level. Is everybody okay with me going to the next slide? I don't see anybody. Okay. So here's what it looks like. This is a good drawing. So there you can see the outer membrane right here. It's kind of been cut away so we can see on the inside. So you can see there's this space between the inner membrane and the outer membrane. And then, of course, we cut into the inner membrane. So there's the very inside of the mitochondria. So I just want to make sure you guys understand what you're looking at there. Again, outer membrane and inner membrane. So there's a space between those two things. And that space is going to be very important moving forward. So make sure you understand that there's a space. Yeah, I was about to say you don't need to know the name of it, but your book is on the same page as me for once. So it just says space between membranes, right? So we don't need to know the name. The fact that there's a space between this outer membrane and this inner membrane is very important, and you'll see why later. What you don't need to know is that this is called the matrix. This is called cristae, right? I'm not going to show you a picture of a mitochondria and ask you to name the parts. I'm not going to do that. But what is very important later is that there's this space between the membranes. That's where a lot of the magic happens. It's not really magic, but you know what I mean. So any questions about this picture? Anyway, I forgot to say, this is a drawing, right? So this is a good conceptualized idea of what it looks like. And this is actually a scanning electron microscope picture of one. So that is what one actually looks like. That one's obviously from 1945 because it doesn't have color. I'm making that up too. They didn't have those scanning electron microscopes back then. But yes, it is in black and white. Anyway, let's move forward. So before we talk about exactly how those proteins work, I want you to think about this. The energy stored by the electron transport is something like the water behind a dam. So before I get into these bullet points, let me just go ahead and tell you. I'll try to draw it, but it probably won't work out for me. Those little proteins, which you can't see, but I've already told you, right? They're embedded on the inner membrane. So you have to imagine there's just a bunch of proteins right here embedded on the inner membrane. And what they're doing, as you're going to learn later, is they're pumping hydrogen ions. So they're pumping these hydrogen ions that are inside the membrane. They're pumping them out in between the inner membrane and the outer membrane, right? So a bunch of pumps, basically. That's what's happening. They're pumping it. These proteins are basically pumps. So that's what I mean by comparing it to a dam. Any of you know how a hydroelectric dam works? And I know at least one of you does because one of you's had my Bio 108 class. So this is very basic. We have water on the other side of this dam, right? There's water over here that you can't see in the picture. And which way does the water want to go? Does it want to go this way, or does it want to go left or right? Left, right? It wants to go left. Why? Because of a force called gravity, right? Gravity is trying to say, hey, you want to go this way. But we have this dam here holding it back. So what happens is they'll let it open every once in a while. The water comes rushing through with a lot of force, and that turns a turbine, and that turbine makes electricity. In the same way, a mitochondria kind of works, except it's not a river flowing in and we dam up the hydrogen ion, but we still are pumping hydrogen ions from the inside of this membrane, the most inside of the mitochondria, in between those two membranes. And it is, that inner membrane is in like a dam. It's just holding back those hydrogen ions. And those hydrogen ions want to flow back in here. Now, it's not gravity that's doing that, but all the stuff that you learned in chapter 5 that I told you was going to be important later, it's diffusion, right? So we're going to have an area of high concentration. There's going to be a lot of hydrogen ions between these things, and they're going to want to diffuse back down, but they can't. They can't go through the membrane. So what happens? And we'll talk about what happens here in a second. But again, for now, you should just think of the hydrogen ions in between these two membranes are basically like water stored behind the dam, trying to get back downhill, so to speak. And then once it happens, once they do flow back downhill, it is almost like the turbine in a dam. So the turbine lets the water through, right? Gravity just does the work, pushes the water through, and then that turns the turbine, and that creates electricity. And in this case, we have little enzymes that literally turn like turbines. And what happens is those hydrogen ions just flow down naturally because they're trying to diffuse, and we know diffusion is just a natural process. So those proteins let those hydrogen ions flow through. That turns it, and that produces the ATP. And I don't usually ask you what the name of enzymes are, but this is one that you need to memorize. Those proteins that allow those hydrogens to flow back through and to turn and synthesize the ATP, they're called ATP synthase. So know that one. ATP synthase, those are the enzymes that make the ATP in the electron transport chain. Because glycolysis, and I showed you it doing some work, and I told you you don't know the name of it. That one's relatively unimportant for our conversation. These are very important. ATP synthase, those are the ones making the ATP. And it's in the name, right? I've told you before, enzymes are named after their substrate and or what they do. And they end in ACE, right? So ATP synthase synthesizes ATP. And I would ask you if you have any questions, but first I want to show you a picture that visualizes the last two slides. And then once you've seen it, and if you still have questions, then I'll ask. So here we go. So we have all these electrons, right, that have been stripped away from the first two stages. They finally make it into the mitochondria, to the electron transport chain, and they literally go from protein to protein, right? So this thing drops them off at this protein, and then they go to that one, and they go to that one. We've got some more being added to it. You don't need to know that detail, right? The only detail you need to know is that we've brought high energy electrons to this process. They go from protein to protein. Every time they do that, they're giving off energy. That energy is used to pump hydrogen ions against their concentration gradient. So that's the opposite of diffusion, remember? Because we are going from an area of low concentration to an area of high concentration, right? This is low concentration. This is high concentration. So we know that naturally things want to diffuse this way. So that means it's going to take energy to make it go that way, right? That's facilitated diffusion. Active transport, right? So we need energy to do that. Where is that energy coming from? It's coming from these high energy electrons that have been stripped away from the food, pumping those hydrogen ions. And then again, this is the part that I've already told you about and described, but now you can see it a lot better. Here's the space between the inner membrane and the outer membrane, right? So these hydrogen ions are just stuck there like water behind a dam. And the concentration is really high because we keep pumping them up into there. And then nature takes its course, right? They want to diffuse from here to here. They want to go from an area of high concentration to low concentration. That's just what, like Brownian motion, right? It's nature. So this thing allows it to happen. It opens the gate, they flow through, it spins, that produces ATP. Yeah, that's it. Of course, this picture has a lot more detail than you need to know. You can look it up for know that. That's the names up. I want to look it up. find out. Again, with this, technically, I keep saying that oxygen is the last thing that gets the electrons. That you do need to know. But then, yes, technically, it's mixed with 2 hydrogen ions, and that forms a water molecule. But again, the numbers you don't need to worry about. Hydrogen you don't need to worry about. To throw back to what we were talking about when we first started this lecture today, electrons come from food, right? Come from the glucose. They make their way to the electron transport chain with an ADH and FADH2. They come in. Oxygen ultimately accepts them, and in the process, that produces water. So yes, don't worry about those numbers. Don't worry about the fact that there's 3 proteins. It doesn't matter. You just need to know that there's proteins, and they're pumping hydrogen ions against their concentration gradient, which will eventually come back down their concentration gradient in something called ATP synthase, which obviously produces ATP. There's one more thing. Any idea why they might have this? Why have the skull and crossbones right there? What does that have to do with any of this? It's because pirates don't do cellular respiration. No, that's not true. Okay. Basically, what they're saying here is this is how, like, when you die from suffocation, like when you die from not having oxygen, this is what kills you. Because ultimately, all those electrons that you're stripping away have nowhere to go. Therefore, you can't produce ATP. Therefore, your cells can't function, and therefore you die. So that's what your book is getting at here. This is how a lack of oxygen kills you. Anyway, any questions about this slide? All right, then the next word for attendance, since we're looking at this cool thing, will be Jolly Roger. Jolly Roger. And I guess if you are watching the video, if I don't have questions for the video, not the people live streaming, but people watching the recorded video later today, if you're watching the video, take a screenshot of me pointing at that Jolly Roger. I wish I had no sleeves on. I actually have that tattoo right there. It would look kind of neat. I mean, stupid if you're not into it, but I think it'd be funny to have them both on the screen at the same time. Anyway, let's move forward. So we are done talking about the 3 stages of cellular respiration. As far as the exam is concerned, we're done with the meat and potatoes of chapter 6, because almost all of it is going to be dealing with those 3 stages. Again, just as some examples, which of the 3 stages uses oxygen? Electron transport chain. Pyruvate, glycolysis. Which of the 3 stages occurs inside of the mitochondria? Electron transport chain and citric acid. So that's what most of the exam is going to be about. Keep that in mind. Now let's talk about the results of cellular respiration. As I've already told you, this is one of those few numbers that you need to know. We generate about 32 ATP per molecule of glucose. I'll go ahead and tell you now, I'm not going to try to trick you. There won't be a test question where I say, how many ATP molecules are produced for 4 molecules of glucose, right? That would be tricky. Because then you'd have to do math, and also you'd have to catch the fact that I slid in that whole 4 molecules of glucose. But I'm not going to do that to you. So for studying, you can make a little flashcard and just memorize about 32, about 32, about 32. I'm not going to trick you. Of course, if I do, maybe it'd be a good extra credit question, like in a whole different section, like you finish the exam and it says extra credit, so you know what you get into. Then I might say how many ATP are produced for 10 molecules of glucose. Because that'd be easy math, that'd be 320. But anyway, so the next slide I'm going to show you is going to show like a little flow chart to keep track of the ATP generated, even though we've already talked about it. Now we're like going back to what we talked about, and we're just, where does this happen? Where is this produced? Where is this used, right? We're not going to do that for a second. Also, again, I don't make these memes, I find them. But that's a fun little independent work question if you want to look into it. As we already said, this is a good time to remind you of stuff we talked about way back when we talked about glycolysis. Even though you don't need to know it for the exam, glycolysis itself is broken down into 2 things, 2 stages, 2 phases. There's the energy investment phase and the energy harvesting phase. So to start this whole thing, to kick this whole thing off, we need ATP. Yet we get ATP from this process. So the question is, where did the first ATP come from? So you can look that up for independent work if you want. And it might be a little complicated. So, you know, if you feel overwhelmed, don't feel bad if you just want to paraphrase it and just make it short and sweet. Actually, to me, that's better for a 100 level non-biology majors course. That's what you need to be able to do. Read something long and complicated and then just be able to condense that down into 2 basic sentences. Like, all right, I don't understand most of this, but here's the gist of it, right? That's so much better than giving me some repeating basically what's in some journal, right? I want you to know, I want you to write what you have figured out from that. Whoops. There we go. So again, we're just keeping track of the ATP produced. Again, we have 2 ATP from glycolysis. More importantly, we're stripping away high energy electrons. Then we're getting things ready for the citric acid cycle. And as far as we're concerned, the conversations we've had, whoops. Oh, come on. I didn't mean to get that little green part there, but this is the citric acid cycle, all right? So as far as the exam is concerned, remember, these 2 things are the citric acid cycle, right? Because we keep saying, all right, so the pyruvate has to be groomed to get into the citric acid cycle. And we talked about the lobbing off a carbon and stripping away more high energy electrons and attaching it to the enzyme, right? All that for the exam, we're all considering that citric acid cycle. But anyway, again, produced a little ATP here, stripped away high energy electrons. Produced a little bit of ATP here, stripped away high energy electrons. Those high energy electrons go to the electron transport chain, where the money maker, right, where we make a lot of ATP. So you got about 2 there, about 2 here, about 28 here, for a total of about 32. For the exam, again, I want to remind you, you just need to know this number of about 32. And then the other 2 test questions about ATP are going to be, I've said it a lot, so hopefully nobody misses this. Which of the 3 stages produces ATP? All 3 of them. Which of the 3 stages produces the most ATP? Electron transport chain. And that's it. You don't need to know that this one produces 2, and that one produces 2, and this one produces 28. You need to know they all produce a little, or at least a little, and this one produces a lot. So any questions about that side? All right. So I keep saying glucose, glucose, glucose. And for the exam, focus on glucose, right? Just to keep things simple, we're just focusing on glucose. Where do the electrons come from? They come from the glucose, right? What do we do in glycolysis? We break down glucose to make pyruvate. It's all glucose, glucose, glucose. Glucose, that's it. We just keep talking about glucose, glucose, glucose. But in all actuality, level non-biology major course, so we're focusing on glucose. But that being said, for your own knowledge, right? So if you're watching this, you can just go ahead and turn your brain off of exam mode, because I'm not going to ask you any of this for the exam. This is just side knowledge for your own knowledge. Obviously, we eat stuff other than glucose. How many of you had breakfast today? All right, just one person. Did you have pure glucose for breakfast? Pure glucose? Like, did you have a glucose solution? Yeah, me either. Right? So we eat other things. Throwing it back to chapter 3, we eat lipids. We eat other carbohydrates. We eat proteins, right? So yeah, that's what this is addressing. Respiration is a versatile, right? It's a versatile process. It burns other molecules too, not just glucose. In the next slide, I'm going to show diagrams, some of the metabolic routes for the carbs, fats, and proteins as fuel. So let's talk about that. Oh, and there's another good independent work question. Break them down, we digest them and use them. But we never mention nucleic acids, right? So when you eat plants or animals or fungi, right, when you eat stuff, you are eating DNA and RNA too. So what happens to that? And if you want to look that up, do we digest DNA? Do we digest RNA? Do we use it? Like, how much do we break it down? Do we break that long strand of DNA down to A's, T's, C's, and G's, and then we use those A's, T's, C's, and G's to make our own DNA? Anyway, independent work topic if you want to look into it. But this is where it works. This is a very simplified version. You eat your food. Your food has carbohydrates, fats, and proteins. What happens to the carbohydrates? Well, that's basically glucose. If it's not glucose, it's strands of glucose. So we can break those down to actual glucose. and that goes directly into glycolysis. Fats, as we know, are made up of two parts. The fatty acids, the 3 fatty acids attached to the glycerol. That is broken down when you digest it. The glycerol goes into glycolysis. The fatty acids go into the pre-citric acid cycle. Proteins, as we know, there's 20 different proteins, all these different amino acids, and they're all so different because they have the different side chains. Some of them are polar, some of them are non-polar, all those different proteins, right? All those different amino acids. And those themselves are broken down. And of course, depending on which amino acid it is, they might go into glycolysis, they might go into pre-citric acid cycle, or they might go directly into the citric acid cycle. And guess what? You don't need to know any of this for the exam. It's just showing you that, we eat stuff other than glucose, and that stuff is broken down. But if I do, I will give you this exact figure. I'll ask you about one of those things. I'll say, which stage are fats processed, right? You can just look at the picture and say, okay, well, fats are broken down into glycerol and fatty acids, and that is glycolysis and the pre-citric acid cycle. Or as far as we're concerned, like I already said earlier, we consider this the citric acid cycle for the exam purposes. But again, 99% chance I won't ask you that. 99% chance I won't ask you that. And if I do, I'll show you the picture. And actually, come to think of it, I won't ask you about fatty acids because, that's a little bit tricky. Is that citric acid cycle or is it not? I would probably only ask you about proteins or sugars. But anyway, let's move forward. Unless there's any questions about this. So again, notice that there's no DNA up there, right? Are we not breaking down DNA when we eat it? What happens? We just pooping out full strands of DNA from the plants and animals and proteins that we eat. All right, here we go. The fun part. Fermentation. So as far as the exam is concerned, this is really like if the size of the font was showing the importance of this as far as the exam is concerned, then this will be really large. The middle one would be really large. The top one would be smaller than that one, and then this one would be even smaller. You might have like 2 questions about the exam. But let's talk about it. As I showed you earlier when I showed you that skull and crossbones, or the Jolly Roger as I called it, because that's what you call the pirate flag. Anyway, when I showed you that, I said that's how people die, right? That's what kills you when you don't have oxygen, is because your electrons have nowhere to go. Therefore, you can't produce ATP. Therefore, your cells die, right? But your book does point out that your cells, and I'm talking about humans here because like bacteria cells, they can work just fine without all this. And I'll tell you about that later. But for humans, yes, we can work short periods without oxygen. Like how many of you have ever held your breath? Yeah, me too. Especially as a kid, growing up in the South, swimming all the time. We used to do it all the time. I never died, at least not from that. Did any of you die? Nope, there you go, right? So there we go. Living proof, you can hold your breath. You can go without oxygen and still survive. Or even if you're running a lot, when you're out of breath, in a sense, you're not completely out of oxygen, but you're relatively out of oxygen, but you survive it. And that's where fermentation comes in. And you need to know, and maybe you need to know the definition of fermentation. Probably not. I'll probably just ask you a couple questions about the process. But fermentation is the anaerobic harvest. We're stripping away those high energy electrons, except this time we're doing it without oxygen. And again, I don't come up with these memes. I guess I need to get rid of that. I've been doing this so long. That's probably not even relevant anymore. Does anybody even know what that refers to, that meme? Not the biology part, but the cultural thing. Well, Taylor Swift was getting an award, and Kanye West busted out on the stage and interrupted it. Said, I'm going to let you finish, but I got to tell you, so-and-so should have won the award. And I've got to update my memes. You didn't even know that? It's killing me. Does anybody not? You guys not like Taylor Swift? You don't like Taylor. You need to know her history. You can't just like her new stuff. You got to know where she came from. Romeo and Juliet and all that stuff. Anyway, let's talk about it. We're going to break fermentation down into 2 basic types. First of all, we're going to talk about fermentation in human muscle cells. Obviously, that's going to be relevant because we are humans and have muscles and have muscle cells. And then we're going to talk about fermentation in microorganisms, which to me is the more fun stuff. Because when you're not in biology class and someone asks you about fermentation, usually what they think about is booze, I think. When I ask people about fermentation, I mean, yeah, I'm working on my moonshine. That's fermentation, right? And that's fermentation in microorganisms. Here we go. Fermentation in human muscle cells. After functioning anaerobically or without oxygen for about 15 seconds. Muscle cells generate ATP by fermentation. You don't need to know that number. What I'm basically, depending on how in shape you are, this respiration that I just taught you, and you start doing fermentation. Why is that? Because of something that you might need to know for the exam, which is this bullet point. Fermentation relies on glycolysis only. So do know that of the 3 stages, which of the 3 stages does fermentation use? And it's only glycolysis. Why is that? Well, simply because glycolysis does not require oxygen directly, which is good, right? Because that will be a test question. Not about fermentation, but when we're talking about the whole process, back it up again, that's going to be a test question. Which of the 3 stages uses oxygen? The correct answer, the most correct answer, is electron transport chain. Of course, you could argue glycolysis and citric acid use it indirectly, but the ETC uses it directly. Anyway, so again, glycolysis does not require oxygen directly. As we already know, it does produce about 2 ATP. I put the star there because it's about, right, per glucose broken down into pyruvate. And you don't need to know that. So these, you don't need to know those 2 for the exam. This bullet point here will just help you memorize. It'll help you remember the one thing you do need to know, which is that fermentation uses glycolysis. And if you can remember that fermentation is a process that happens because you don't have oxygen, and you can remember that glycolysis doesn't require oxygen, hopefully that'll help you remember those 2 things. But other than that, I don't require you to know that glycolysis doesn't use oxygen. That tidbit is there to help you. Anyway, any questions so far? We're going to talk about this some more. All right. So again, it's all about stripping away the high energy electrons, right? And what does the stripping away? It's this thing, right? So remember NAD+, that's the thing. When it has a plus, technically it's still waiting to pick up the electrons. It's not NADH until it gets those electrons, right? So we need a lot of NAD+, right? To strip away high energy electrons, we need the stuff that strips them away, right? And when you have regular cellular respiration, right, in aerobic conditions, when you have oxygen, that's not a problem because as we know, those high energy electrons are stripped away. They're taken to the electron transport chain. NAD+ ready to go again, right? Because it's basically, like I said earlier, they're basically like buckets. They hold electrons. So when they're full, there's nothing they can do, but then they drop them off. Then it's an empty bucket again. They go back. Process continues, right? Fill them up, dump them off. Fill them up, dump them off. That's what they do in regular aerobic conditions where you have oxygen. But what happens when you don't have oxygen? You have no place to dump the electrons, right? So you have this full bucket, and then all your buckets are getting filled up because you have nowhere to dump your electrons. So therefore, if you have all your buckets filled up, then you have no way to strip off the new electrons. Therefore, you can't make ATP, and that's an issue. So that's what this problem, that's what we're talking about here. And now we don't have the oxygen, therefore there's nowhere to go. Therefore, you can't dump that bucket and you can't get back to the empty bucket. You've got the full bucket. So you can't strip away electrons. And if you can't strip away electrons, you can't make ATP. Which is basically what this is all saying, right? So everything I just said is right here. Now it's in writing. There's no oxygen to accept those electrons. Oh, here's the new one. Let me draw a little dashed line here. There is new information here. So electrons taken. What happens to them? Back to the pyruvate instead, right? So we started with glucose. We broke it in half to make pyruvate. In the process, we stripped off those high energy electrons. Now those high energy, we're just like, all right, pyruvate, you need to take these electrons back. We have nowhere to put them, right? So you dump your bucket, right? You get rid of your electrons. So now you've got the empty bucket again, the NAD+. It's ready to do its job. It's ready to strip away more high energy electrons. So then glycolysis can keep going. So for the exam, I would say the more important bullet points are this one. Where do the electrons go? They go back to the pyruvate. And then what does that produce? Right here. That's the other important, probably as far as the exam is concerned, the most important, like in that it's most likely going to be the question. So anyway, those electrons are added back to the pyruvate, and that forms lactic acid. So most likely, I'm not even going to ask you how lactic acid is produced. I'm probably not even going to ask you to memorize the fact that the electrons have been added to the pyruvate, and that produces lactic acid. More than likely, the question is going to be, in human muscle cells, fermentation produces what? And the answer is lactic acid. Because as you're going to see later when we talk about microorganisms, fermentation can also produce other things. But for human muscle cells, we produce lactic acid. How that works. But again, fermentation in human muscle cells produces lactic acid. And a lot of that is what makes you sore. I don't want to say sore, because you'll see in this textbook, actually, there's other things that make you sore from working out. But if you've ever been running or done anything where you really that uses a lot of wind, a lot of breathing, and you feel a burn, whether it be in your muscles or your chest or whatever, that's usually what that is. It's burning because it's literally acid. Because you're using so much oxygen that you can't, you're not bringing enough oxygen in. So even though you're not holding your breath, you have a relative lack of oxygen. So you're doing fermentation, right? So you're producing lactic acid, and that's what the burn is. Anyway, any questions about this slide? It's all about those electrons. Oh yeah, and there's a visualization of what I was saying, right? So this person's not checking their shave job to see how smooth the leg is. They're having a cramp. They're feeling a burn because they've been running, I assume, sprinting, and they didn't have enough oxygen. Therefore, they were doing fermentation. Therefore, they were producing lactic acid, and it burns them. But I'm not going to ask you about that. That's for your own knowledge. Here's the stuff you need to know, right? Fermentation uses glucose, which is what we're seeing here. We break down this glucose, glycolysis. It uses the process of glycolysis, which breaks down glucose in the pyruvate. Again, just as usual, we strip away high energy electrons. Usually they go to oxygen, but there is no oxygen, so we've got to dump those electrons off somewhere. Where do we dump them? We dump them back to the pyruvate, and that produces lactic acid. So again, you don't need to know the numbers. You don't need to know that for every 1 molecule of glucose, we're producing 2 molecules of ATP, and we're technically stripping away 2 electrons. And then those 2 electrons are then added to the 2 molecules of pyruvate, which gives us 2 molecules of lactic acid. Don't worry about the numbers. Just know that we're breaking down glucose, we're stripping away electrons, we're producing ATP. Those electrons have to go somewhere. And that burns you. So any questions about that? All right. If you download the PowerPoint, you can see this little animation that shows basically everything I've just shown you. But it's animated, so hopefully maybe easier to follow. You do that if you want. Let me think. For extra credit, since the people who watch the videos very rarely get much extra credit opportunities. If you're watching the video, not live stream, if you're watching this video later today or this weekend or whenever, if you watch this video, if you do download the PowerPoint and click on that watch video and take a screenshot that shows that you watched it, you'll get a little bit of extra credit for your attendance. Anyway, every chapter that we go through has this, the process of science. As always, I'm going to skip it because we don't have time for it. As always, I recommend you reading it. It is decent information. The whole idea of the scientific process is very important. You do need to know the scientific process. If anything, for this whole entire course, knowing the scientific process is probably more important than any of the little things I'm teaching you. But anyway, read it. Let me know if you have questions. There's probably questions about it in the study guide. No, there's no questions about it in the study guide or the exam. Yeah. Okay. But if you're a student athlete, this should be interesting to you because what causes the muscle burn, right? So if you're an athlete, that might be relevant to you. Anyway, microorganisms. So we know what happens in human muscle cells. Let's talk about microorganisms really quickly. We can knock this out and finish chapter 6 this week. So again, humans, we need a lot of ATP. We're big, we're complex, so we have to do the full cellular respiration as I taught it. Some things can just do fermentation only, right? For some things, like if you're a single-celled organism, you don't need a bunch of ATP. This is what this bullet point is, right? Fermentation can sustain many microorganisms. Yeast is a good example, and you don't need to know, but you need to know, or you don't need to know, but for your own knowledge, yeast is a single-celled organism, but it's a eukaryote, meaning it's the more complicated, like humans and plants and fungi. It's more complicated, it's bigger. So even though it's single-celled, it's not as simple and small as a bacteria. So therefore, yeast can do lactic acid, which is what we do, right? That's what we make. It can make lactic acid, and it can make alcohol, as I'm sure most of you know, the alcohol part. I think most people think of yeast and think of alcohol. But really quickly, let's talk about the lactic acid. So the lactic acid from yeast is used to make things like cheese, sour cream, yogurt, soy sauce, bacon, cabbage, olives, sausage, meat products, all that kind of stuff. Good independent work topic if you're interested in food. So you can look that up. You know, what do we use fermentation for other than, you know, making booze, right? And here's some of them. And if you're really interested, you can look up how. Like I've got a pressure cooker, and I haven't done it yet, but there's instructions that say how you can make yogurt using the pressure cooker. So you could look into it. I don't know. I mean, don't give me a recipe, but you can look up and explain how it works on the cellular level. But anyway, yeah, yeast does make lactic acid. It also, however, if you starve it long enough and give it the right conditions, it will also make ethyl alcohol, right? So booze. Ethyl alcohol is the stuff, or ethanol, either one. Same thing, different name. That's the stuff that you have in wine and beer and liquor. So that's why you need yeast to. Yeah, and I'm not going to ask you any of that either. Just know in the back of your mind, again, yeast can make lactic acid. It can also make alcohol. And also, for your own knowledge, when you make alcohol, you also make carbon dioxide, which is where the bubbles come from, right? And we'll talk about that here in a second. Even though you don't necessarily need to know it at that level. There's some pictures of some yeast. That's a bunch of different yeast because, again, each one of these little things is a yeast, right? And for your own knowledge, yeast is in the fungi family. Anyway, this should look really familiar. This looks a lot like the fermentation I showed you in human muscle cells. We start with glycolysis. The whole thing is glycolysis, the process. We start with glucose. We break it down into pyruvate. We're stripping away high-energy electrons. Those high-energy electrons don't have anywhere to go. Right? The bucket's full. We can't dump them. So where do they go instead? They go back to the pyruvate. But in this case, instead of going directly to the pyruvate, in this case, we've lobbed off a carbon from each one of them. Therefore, we produce some carbon dioxide. And therefore, now we have these 2 carbon molecules instead of the 3 carbon molecules of lactic acid. Not that you need to know that detail for the exam. But here we go. Instead of lactic acid, we get alcohol. So it's the same thing, except we're producing alcohol instead of lactic acid. So for the test, the question is going to be something like, human cells produce blank, while yeast cells can produce blank, right? So obviously, if you were to say lactic acid and lactic acid, that would be a correct answer because, again, yeast can do both. But the most correct answer is human cells produce lactic acid and yeast cells produce alcohol. For the test, we're going to focus on the fact that they produce alcohol. Yes, again, they can make lactic acid, but yeast can produce alcohol, human cells can't. Can you imagine if human cells could produce alcohol from the same thing that causes us to produce lactic acid? You just see some drunk dude on the side of the road, like, what happened to him? Well, it's like, well, he went for a sprint. He went for a run and was sprinting too much, got out of breath, and here's a bunch of alcohol, and I was drunk. That would be horrible. That's fermentation that produces alcohol. Any questions about that? All right. Actually, before I go forward, let's see. Okay, then the next word, or I guess the last word for attendance will be jogging. Since I'm talking about somebody going for a jog and getting drunk because their body produces alcohol in an alternate universe. Anyway, that's the last word for attendance is jogging, because here we get to the evolution connection, which I skip. We have every chapter and I skip it. So read about it. Ask me questions if you want. It is really interesting, but it's nothing that I'm going to test you on. Basically, we're showing, You just read it. I won't waste your time. It's Friday. So that's it. And then also, if you download the PowerPoint, you can watch a review. So if you're studying, you can go back through this and look at the review, but I'm not going to waste your time doing that. So that's it. We are done for today. Make sure your pre-labs are done before Sunday. Remember, you don't have to do them alone, especially this pre-lab, because next lab is so important. So come to me if you need help. And have a good weekend. And if you're online, don't forget, send me those words before 9 a.m. Anything after that, and you're late.